Hello, I'm Dwayne Birde. My roll number is 6207, and the enzyme of choice for me is Cas9 nuclease. Let me give you a brief introduction about what Cas9 is and what it has relation to CRISPR. So, Cas9 is an enzyme. It is a single effector protein nuclease enzyme that actually is a nuclease enzyme that cuts the cuts the DNA by causing double-stranded breaks, the DSBs. So this enzyme is unlike any other normal nuclease because it is a very special kind of enzyme. The thing that makes it special is that it is guided by an RNA molecule. Never ever have we heard of an enzyme that is guided by another enzyme. It is actually guided by itself with the help of some cofactors and all that. But this enzyme, the Cas9 nuclease enzyme, is actually guided by an RNA. That RNA molecule is called as gRNA. So this uh, Cas9 nuclease enzyme comes with actually a complex is seen in the form of a complex rather than as an individual entity. So that complex is actually the the CRISPR RNA, CRISPR, I mean the guide RNA, single guide RNA, Cas9 complex. So Cas stands for CRISPR associated nuclease, CRISPR associated nuclease, and this enzyme is widely known today and was very famous for its use in this technology, in genome editing technology. So this was just a brief introduction about the Cas9 nuclease. Let us delve back to the time when this enzyme actually began showing its appearance in the scientific community, and when it was actually discovered, and why is it so famous today for the genome editing technology. So let's begin with its history here. Uh, this enzyme was discovered in 1987 by a Japanese scientist Ishino and in the well-known E. coli bacterium. So, uh, to give you a little more introduction or info about why it was discovered and how it was discovered. So, this scientist Ishino was studying the conversion of alkaline phosphatase enzyme. So, um, into some other form in the periplasm, and so for that he wanted to know the sequence of the entire genome of E. coli. So as he sequenced, he noticed that there were some sequences that regularly appeared on the genome of the bacteria, and these sequences, not then but later, came to be known as CRISPR. What does it stand for? It stands for clustered regularly interspaced or interspersed short palindromic repeats. So this uh, enzyme or the CRISPR repeats are actually found in prokaryotes as well as eukaryotes. Uh, in the case of archaea, it is found in Halopharax mediterranei, first founded by Mojica in 1995 in archaea. So there were many contributors to to the discovery of Cas9 nuclease. I mean, they were not intentionally finding for it, but they eventually got to know, eventually found out certain discoveries that led to the discovery of Cas9 nucleus in the future. So it was first Ishino, as I said, discovered CRISPR repeats in the E. coli genome. Mojica in 1995 discovered uh, same repeats in archaea. And another interesting thing about Mojica's discovery was that he was the first to hypothesize that unusual loci includes fragments of foreign DNA. So let me tell you, just adjacent to those CRISPR repeats exist sequences of foreign DNA. Now, why is that? Let me give you a short introduction about it. That these foreign DNA are just like the immune memory of humans. So Bacteria has evolved and has got an adaptive immune system, which it uses to fight off bacteriophages that infect the bacteria. So once the bacteria infects the bacterium in the first place, it stores the genetic material of the bacteria, and then uses that same genetic material or the same sequence to fight off the bacteria, or the bacteriophage, when it infects again. So it's just like the immune system of our body that has got an immunological memory to fight off an infection when uh, attacked by it again. So moving ahead, Marafini and Sontheimer, they discovered that the target of this nuclease enzyme was DNA, and they talked about its potential for genome editing. Again, in Rudolf Barango and Philip Horvath, they discovered in 2007 the mechanism of action of Cas9 nuclease, which we will be discussing ahead here. They worked on Streptococcus thermophilus while working for the company Danisco, observed the development of acquired immunity upon addition of new spacers. So they experimented and tested if um, Mojica was right in his assumption or hypothesis that this foreign DNA is a part of the immune system of the bacteria. So they actually inserted few sequences that belonged to bacteriophage DNA, and then they saw that the bacteria was able to resist infection by the same bacteria to which the sequences belonged. So moving ahead to the structure of this very special and well-known enzyme of today's generation, that in the Cas9 nuclease has got six domains, or six, like just like we have organs, that it has got six organs or six arms, uh, which are assigned various different functions. So beginning with the first a domain of the enzyme, we have REC1. REC1 is the largest domain, as you can see, and it actually uh, promotes binding of the Cas9 nuclease to the guide RNA. It is essential that the Cas9 nuclease is bound to the guide RNA. Only then will it actually perform its activity of producing or of inducing double-stranded breaks into the DNA. Then comes the bridge helix. This bridge helix is arginine rich, and it's crucial to, this actually contributes to the initiation of cleavage activity of the Cas9 nuclease. The second, or sorry, the third domain that we can talk about here is the PAM interacting domain. Now, let me tell you what this PAM is. As you can see in this image on the top right, there is written PAM. So these are just three nucleotide long sequences on the um, 5' to 3' strand of the DNA, and they stand for protospacer adjacent motif. Now, what are they? Uh, all enzymes require a specific site on which they will alight, and then they will begin their catalytic activity. This PAM sequence is same, just James, same that I talked about. PAM sequence is the sequence on which the Cas9 nuclease is going to bind to, and then uh, actually initiated cleavage activity. Now, how will it bind to is actually with the help of PAM interacting domain. This confers PAM specificity, thus initiating binding of the Cas9 to the target DNA. The fourth and the fifth most important, the domains that we can talk about right now is the HNH domain and RUVC domain. So these two domains are the nuclease domains. These are the catalytically active domains, as they promote the cleavage of the double-stranded DNA, producing double-stranded breaks, thus helping us in genome editing further. So HNH and RUVC are the nuclease domains. Moving ahead, we have the last domain, which is the REC2 domain. The function of this domain is actually yet unknown. Moving ahead, we have we can talk about the industries that produce the Cas9 nucleus or the CRISPR-Cas systems, I should say, and not Cas9 nucleus, because Cas9 nucleus does not exist as a single entity. So there are many CRISPR-Cas systems that companies synthesize based on their target or the aim. So there are many companies which have the aim of curing blood disorders, which have the aim of doing research in plant genetics, or they have the aim of curing cancer with the help of gene editing technology. So one of the company is the CRISPR Therapeutics. It has been actually founded by the Nobel laureate Jennifer Doudna, and she was actually then, yes, awarded the Nobel Prize, one of the contributors to the CRISPR-Cas systems understanding. So the company's aim here is to cure thalassemia and sickle cell anemia, all the blood disorders. One of the well-known therapy that this company has actually developed is the CTX001 therapy. The another company that we can talk about is the Intellia Therapeutics. So this therapeutics company is again a CRISPR-based company delivering that actually aims to like cure diseases like transerythrin amyloidosis and hereditary angioedema, and it is aiming to deliver this CRISPR-Cas9 system into the body of the humans via lipid nanoparticles. So this is actually a technology, and one of the main challenges of CRISPR is the delivery. How is it going to be delivered into the body? So this company is actually working on that. The next company then we can talk about is the Caribou Biosciences. Caribou Biosciences aims at uh, modification of the T cells, just a minute, modification of the T cells to actually develop therapies that enhance the cancer treatment. So most of the cancer treatment, like today, are based on chemotherapy, radiotherapy, but this company is aiming on immunotherapy, wherein the T cells are going to be modified to produce specific T cells and which carries genes that actually enhance the recognition of the T cells uh, of the tumor cells. So it is actually, so one of the actually product of uh, gene edited gene edited T cells is the CAR T cell therapy. So this therapy is actually widely known outside India, and recently one company is actually developing or has actually developed CAR T cell therapy at a low price compared to the price actually exists outside for India, and it is actually a very nice attempt to do so. So moving ahead with the applications of the CRISPR or the Cas9 nuclease enzyme or the CRISPR-Cas nuclease system, the first application can talk about is of genotyping of myco, uh, Mycobacterium tuberculosis and Aspiogenes. Genotyping itself means that, genotyping means that you are actually differentiating the strains of bacteria by knowing uh, the small differences in the nucleotide sequences of the bacteria, and CRISPR actually is helping in that. Actually, this understanding or the application was actually derived and followed way before actually CRISPR was fully understood. Then the main application of CRISPR-Cas includes the genome editing, T cell model. One of the example is interesting example is of T cell modification for cancer treatment. Another great application that possibly could be of CRISPR-Cas9 is as a direct antimicrobial tool. So as we know, the world is now reeling under antimicrobial resistance, possibly new pandemic. So this CRISPR technology can be used to target such antimicrobial genes, and thus helping us curb the problem of like antimicrobial resistance. The fourth application is of transcription regulation. CRISPR has the ability to either knock out, knock down, or activate the expression of the gene. So what that means is, when I say activation, it means uh, enhancing the um, expression of a certain gene. Knockdown means reducing the expression of the gene, and knocking out means completely stopping the expression of the gene by actually removing that gene from the DNA of the organism. So this is what is called as transcriptional regulation. So this is where CRISPR can be used. And the fifth most interesting and the most comforting, I would say, uh, application of CRISPR includes to cure genetic diseases. The CRISPR is actually so precise and specific that it can be really used for curing and deleting all those genes that are responsible for most of the disorders that the world is reeling under today, like Alzheimer's, cystic fibrosis, Parkinson's as well, and blood disorders like I talked about, just thalassemia, sickle cell anemia. So these are really interesting areas where CRISPR has applications in. So one of the application includes repairing the mutations in the CFTR locus to treat cystic fibrosis. Coming to the reaction and mechanism, so Cas9 nuclease, guided by the single guide RNA, is actually uh, going to cleave the phosphodiester bond of the DNA, and a double-stranded break is going to be introduced into the in the DNA. That will result into the fragmentation of the DNA, and post which it can either, you know, I mean, we can insert a gene or just introduce deletion with the help of CRISPR, and the DNA will be then further repaired by two mechanisms that exist naturally in all cells, which includes homology-directed repair and non-homologous end join. So all these repair mechanisms are essential because we want the DNA to be cleaved, repaired back again, because we can't just cleave the DNA and leave it aside. So homology-directed repair is quite precise one, and while NHEJ just introduces insertions or deletions commonly called as or collectively called as indels. So this is how the reaction of this actually happens. Then mechanism, we can the last thing that we can talk about in this poster is of mechanism. Cas9 nuclease is activated first by bringing conformational change in its structure by binding of GRN. So we all know that enzymes have certain structure, and they are not active at all times. Only at certain point, only when they are needed, they are activated with the help of, as seen in the case of other enzymes other than Cas9 nuclease, with the help of cofactors and like cofactors and all that stuff. So in this case, activation of Cas9 nuclease occurs with the help of binding of the guide RNA to the Cas9. Now, the mechanism isn't fully understood, but the scientist Genek and his colleagues, they hypothesized that steric interactions or weak binding protein side chains and RNA bases of the guide RNA induce the change and activate the nuclease enzyme. Post-activation, the Cas randomly searches for target DNA sequence that matches its PAM sequence. So Cas9 has certain affinity for certain PAM sequences with the help of its PAM interacting domain that I explained you before, that it offers specificity towards PAM sequences. So it is going to recognize only certain PAM sequences on the DNA of the target organism. After getting the target sequence, it melts the DNA, and guide RNA pairs with the complementary region of the DNA. So the complementary region of the DNA is the 3' to 5' in direction, while the PAM sequence is in the opposite strand running in the 5' to 3' direction. Once the base pairing between the target DNA and the guide RNA successfully occurs, The RUVC and HNH nuclease domains cut the target cut the target DNA a slight little distance apart from the PAM sequence, which is actually approximately three nucleotides upstream of the PAM sequence. And yeah, I mean upstream, they cut the Cas nuclease cuts the DNA upstream of the PAM sequence, and thus the double-stranded break is actually introduced. So this was all about Cas9 nuclease, or I can say I should not suggest Cas9 nuclease. It should be CRISPR Cas9 nuclease. A very wonderful technology, booming technology today. And I hope you all liked my posture. And thank you for that. Thank you for listening to me. Thank you.